So these are the sort of data that we're familiar with in publications as we flick through the pages of our journals. It doesn't matter what they are. You'll see that you're seeing one compared to another within each plot there, but you have no idea which of these relates to each other in terms of interactions. This study is a pilot study that was conducted before the full Biomarkers study was conducted, and demonstrates the multitudinous data showing differences in the different biomarkers. But capturing that required a different technology. And this is where the mathematicians came in. And what they did is enable us to free the numerical information from its sites of origin. In other words, to allow the data to function independently on its own within a domain. And this is so-called topological analysis, discovered first of all in the 1700s with those pretty diagrams that you can see there. Let me explain a little bit how you set about doing this. It's a way of using geometry to capture interactions, free of metric space and free of coordinates. Now you can't read this, but this is the London Underground. And you'll see the River Thames there marked in blue, and if you're walking around London, you'll be able to find where you were by looking at the Thames or the Houses of Parliament or Buckingham Palace, and that would be easy. You'll be able to find the tube stations, underground stations, using that as well. But go down the stairs and go underground, and you're confronted by this. And yet, you're still able to get around, because you can look at the relationship between one station to another, connected by those coloured lines. And you can easily negotiate, well, some people can easily negotiate the underground in that way. So this is what one can do for complex data. Over 300 different sets of data were gathered in the Biomarkers study. And one is able to use this by computer binning and sort it into its different associations. We call them handprints, and then begin to separate those to see how they relate to each other and construct the clustering network. So this was the first pilot study. It was conducted on asthmatic patients in the southwest of England. As you can see, there are several different collections of asthmatic patients showing different subtypes of the disease. The healthy normals are shown in blue there, and as we go around this topography map, we see different subtypes of asthma separating quite cleanly from each other. And each of these, of course, have different characteristics that enable that subtype to be differentiated from another. This actually is looking at severity of asthma in red, and you can see that subtypes 5 and 6 particularly contain most of the severe asthmatics, whereas the other ones are in some of the other subtypes. And then we can start looking at the other characteristics that make up that subtype, and you won't be able to read all of this, and it doesn't really matter for the sake of demonstrating it, but you can see that each of these subtypes has very particular clinical characteristics, pathological characteristics, physiological characteristics, which is helpful if we're trying to unpick the complexity of asthma. We can go one step further. We can start applying biomarkers. Let's look at mast cell biomarkers, mast cell tryptase or chymase or carboxypeptidase A. And you can see that there's a cluster there in red and one at the bottom, which particularly picks up the mast cell mediators, whereas the other ones don't. If we now look, for example, at Th2 cytokines, you'll see only one of the clusters is heavily influenced by Th2 cytokines, although there is a little in the one expressing mast cell activity as well at the bottom. And finally, if we go to innate T cells, we see other subtypes.